Good afternoon, everybody. Friends, we want to study here the topic of 12th biology, that is, paper first, the gene, its nature, expression, and regulation. We are discussing about this topic. Now, you know that the genes is nothing but the small part of a DNA, or we can say a segment of a DNA which used in inheritance and heredity. Now that gene, how it inherited or how the characters are transmitted, here explained initially introductory in this topic, so that first of all the genes concept we want to study in the form of DNA. The DNA, most of the scientists proved that DNA is the genetic material in near about of all the organisms, 99% organisms, the DNA is the genetic material. Few of the viruses only have the RNA as the genetic material. Now, is the DNA genetic material or not? For that, initially we want to study the experiments performed by the variety of the scientists. The scientist first experiment, that is DNA as a genetic material, that is Griffith's experiment, we want to study here. Griffith's experiment. In explaining whether the DNA or something is a principle which is transforming the one form of the bacterium to the another form. For that, Griffith chose the mice for his experiment. He performed the experiment on the mice so that the experiment belongs to the mammalian. The disease pneumonia is caused by the bacteria, the Diplococcus pneumoniae. The bacterium causing the pneumonia in mammals. So that Griffith first studied the bacterium Diplococcus pneumoniae, how is causing the pneumonia. Now he studied the Diplococcus pneumoniae. At that time he found that there were two strains of the Diplococcus pneumoniae, that is S-type strain and R-type strain. S means smooth or the virulent. S-type strain of the bacterium causes pneumonia, whereas the rough strain, it is rough type of a strain of the bacterium which is not having the polysaccharide coat, that is, so that the nature of the bacterium is becoming rough. So such R type of the strain is non-pathogenic or not causing the pneumonia, that is avirulent. This is virulent strain and this is avirulent. Means due to R type, the bacterium is not causing pneumonia in mammals. When he performed the experiment, Griffith first injected the strain of the bacterium, that is S-type strain of the bacterium, into the healthy mouse. He found that the mouse after some days suffered from the pneumonia and finally died. Okay, means the S-type strain is pathogenic strain causing the disease and the mouse will be die. On the other hand, R type of the strain is when injected to healthy mouse by the Griffith. He found that the pneumonia is not caused, remains alive. This mouse remains alive, so that the R type strain is non-pathogenic. Such two steps first he made, and in the third step what he done, S type strain of the bacterium get heat killed. Griffith taken this S type, this pathogenic bacterium into a flask and then kill this S type bacterium by heating it and then get into injection syringe and then injected to the healthy mouse. Then heat killed strain S type when injected, the mouse remains alive in the third step and it is also non-pathogenic. This is the third step made by the Griffith. At the fourth step, what he done, see, that is very interesting one. S-type strain, S-type heat killed plus R-type get mixed by the Griffith in the same injection and injected into healthy mouse. At that time he get surprised after the result because the mouse will suffer from the pneumonia and finally die. Griffith wondered how this is possible. He just thinking about that. He thought that when R-type strain separately injected, the mouse not suffer and remains alive. When S-type heat killed strain get injected, mouse not suffered, remains mouse alive. But what happened when the mixture of S-type heat killed and R-type alive strain mixed and injected, how the mouse will be die? He finally get conclusion, something is there which is going to transforming the one form of the strain into another form, and that which is going to transforming, that is called Griffith as a transforming principle. Griffith not perfectly given the solution what is the transforming principle, but he told that in this mixture the change will be observed and the mouse get suffered and due to pneumonia and finally died, and he concluded that something is there which is going to transforming from transforming the one of the strain of the bacterium into another form, and that transforming principle something is there. First of all Griffith concluded, and finally he cultured the blood samples of the dead mouse of the fourth set, and finally when observed, he observed the colonies of S-type bacteria. S-type bacterium, that is pathogenic bacterium colonies, are observed by the Griffith in this set, which is not injected, and Griffith wondered how these colonies will be developed and finally concluded that something is responsible to convert the S-type strain, and that converting something is called as a transforming principle. Now S-type in the fourth step, S-type heat killed get converted into S-type by the using R-type. Now R-type strain get the surrounding nutrient medium is S-type, and R-type get converted into S-type, and that is due to S-type heat killed. This is the something which is Griffith stated in his principle, in his transformation. Now, one form of the strain, that is R-type strain, get converted or transformed into S-type, and finally the Griffith gives is a conclusion that something principle. Afterward, later on the Griffith's experiment, or after the Griffith experiment, other scientists told it is nothing but the DNA. Now, in support the Griffith's experiment, the another experiment made by the three scientists, that is Avery, MacLeod and McCullough. Now these three scientists, after 16 years by the Griffith performed his experiment in 1928, and in 1944 they performed, these three scientists of different places now performed the experiment in support of the Griffith's experiment, and they proved that DNA is the genetic material. What they done? They obtained the S-type, which are obtained in the fourth set by to the Griffith. That strain is get isolated from this fourth set of the Griffiths. Now they made a test tube assay for this experiment. In the test tube, first of all, they got the nutrient medium, that is R-type bacterium, in the test tube. The test tube assay made by these three scientists in support of this experiment. The culture medium, surrounding to this R-type bacterium, R-type strain of the bacterium. Here, first of all, taken as a surrounding medium, that is heat killed S-type, and in that R-type bacterium is introduced. That is, in each test tube, these three scientists taken the chemical components of the S-type. S-type get isolated into separate components, that is, the carbohydrate is get separated from S-type, carbohydrates, then proteins, then DNA, and then DNA plus DNA's enzyme is added. Now, see, whatever the test tube assay, here each component of the S-type is get separated and separately taken into separate test tube first of all. Then they added the R-type strain surrounding with the culture medium, which is consisting the heat killed S-type, that is carbohydrate gets separated, proteins get separated, DNA gets separated, and DNA plus DNase enzyme, which is nothing but the actually DNA is going to digesting by the DNase enzyme in the last step. He observed that nobody except one case, nobody will be changing this rough type of the bacterium, R type of bacterium into smooth. That is, when he observed, the scientists observed the carbohydrate, whether the carbohydrate is responsible to changing this R type into S type, no result. So that remains as it is R type. Here, the second test tube, when proteins are separately filled into this test tube by isolating S type, then here also the proteins are not changing the R type into S type. But in the third step, in the DNA test tube, the DNA, the R type get here changed into S type. And finally, in the last test tube, DNA is going to destroying or digesting by enzyme, DNA plus DNase enzyme. There also the R type strand remains as it is. It means that the changing principle, transforming principle of R type into S type is nothing but the DNA. In this way, these three scientists, in support to the Griffith, proved that DNA is the genetic material. DNA is the transforming principle of the Griffith. But even though these scientists proved, all the scientists, all the biologists not accepting or not convinced to this experiment, they are not accepting, and again final experiment that is made in support to this Griffith experiment, that is for the DNA as the genetic material. The last experiment made by the Harsha and Chich. Third experiment, Harsha and Cheese experiment. What they made, actually these two scientists are the studying on the viruses. The viruses, those are infecting the bacteria. The viruses, those infecting the bacteria, they are said to be bacteriophages. Now, for the experiment, they used the bacterium E. coli, Escherichia coli, which is going to be infected by these viruses. And first of all, E. coli bacterium taken, and for that they used whether the genetic material is DNA or protein. In these two, definitely there is a genetic material, so that they used the radioactive isotopes of phosphorus and sulfur. That is phosphorus, that is radioactive phosphorus means P32, and sulfur radioactive, that is P35, that is radioactive sulfur and radioactive phosphorus. These two radioactive substances used by the scientists to prove whether the DNA is genetic material or not, or protein. Why they are taken these radioactive isotopes? Because you know that the DNA has the phosphorus but not sulfur, whereas proteins has sulfur but not phosphorus. So that they taken first of all the radioactive phosphorus and sulfur in their experiment, and they introduced this radioactive phosphorus as well as radioactive sulfur into the bacteria. They observed that the protein coat, actually the bacteriophages has two chemical components. The centrally situated in the head region, the bacteriophage, as we know that the bacterium, suppose this is a bacterium, and here in the head region the bacterium has DNA, and this is the core or coat is nothing but the protein. Bacterial cell, E. coli bacteria, when some of the viruses are taken or as a isotopes or introduced isotopes of phosphorus and some of the viruses are introduced into viruses introduced by the isotopes of the sulfur. Now when these few viruses of sulfur isotopes and few viruses with phosphorus isotopes are introduced or infected to the bacterial cell, they found that the final, when the bacterial cell infected by these viruses, which are some of the viruses with the sulfur and some of the viruses with the phosphorus, only the DNA of both the viruses are introduced into the bacterial cell, and this protein coat remains outside. Only DNA. Now here the DNA of these viruses is isotopes of sulfur, that is 35, and this suppose isotope of phosphorus. Now these viruses are introduced with isotopes of sulfur and these viruses introduced with the isotopes of phosphorus. Only DNA is introduced into the bacterial cell. The protein coat remains outside of the bacterial cell, and finally the E. coli bacterium is finally observed that the E. coli bacterium, where introduction of isotope of phosphorus, that is DNA, that is radioactive, but here sulfur is not radioactive. And finally, the radioactive phosphorus, here the DNA is introduced in the bacterial cell, that is infecting to the bacterial cell, resulting, indicating that DNA is the genetic or inheriting material, and that is they proved the DNA as the genetic material. In this way, we first studied that DNA as the genetic material. Different scientists made their explanation in the experiments and finally concluded that DNA is the genetic material. Friends, now we concluded that DNA is the genetic material. DNA, the deoxyribose nucleic acid, is the genetic material. Few of the viruses have RNA is the genetic material. After that, we want to study the second point, that is modern concept of the gene. In this as we know that gene is the segment of DNA useful for the inheritance, that is changing the character from one generation to another, that is the small fraction of the DNA is nothing but the gene. In modern concept, the Seymour-Benzer given the three terms in the modern concept of the gene, that is first cistron, the muton and recon. Resistron, the unit of function, the muton, the unit of mutation, and recon is the unit of recombination. Such three concepts as a modern concepts of the gene were given by the Seymour-Benzer, which are having different lengths in variation, that is unit of function. Cystron has few hundred nucleotides base pair long. The muton is the one to few nucleotides long, whereas recon is the one few nucleotides long. Such new concept of the gene given by the Seymour-Benzer. That's about the concept of the gene as a modern one given by the Seymour-Benzer in 1955.